So the repressor is always on the lac operon. It's inactive until lactose has entered, and then the repressor goes off because we need to make protein so that the protein can fight the lactose in our body. Operon is always on unless you eat something that has tryptophan in it, so then the repressor would attach so that you would stop making proteins because you don't want too much in your body. The lac operon is always off, which means the repressor is always on it. So it's mostly inactive unless we eat something that's lactose, then the repressor then goes off, so then we can make those proteins. The tryptophan or the trip operon is always on unless we eat something that has tryptophan in it, which would make the repressor attached to it, so then we don't have too much in our body.
The product that comes from both of these is that the repressor helps to limit the amount of lactose and tryptophan that is produced and keep the levels high enough or low enough and just at the right amount so the body does not awkwardly function. In cell division is simpler in prokaryotes rather than eukaryotes due to the same thing as replication of DNA. Prokaryotes are just base simpler and don't have as many different complexes to them and are a lot simpler, so it occurs a lot faster and is a lot more basic. In gene regulation, for the prokaryotic cells, it occurs in the transcription level, and DNA is found in the cytoplasm, but for the eukaryotic cells, it happens at many different levels, and DNA is found in the nuclear department. So for replication of DNA, the biggest difference is based on size, and complexity is the biggest difference because since prokaryotes are more easily set up and simply based, it's a lot easier and it's not as much that needs to be done, so it happens at a faster rate. But for eukaryotes, since they're very complex and have a lot of different parts to them, it takes longer and is happening in many more steps. For transcription and translation. For prokaryotes, it happens at the same exact time and happens simultaneously. But for the eukaryotes, the RNA is translated first in the cytoplasm, and then transcription occurs. So for prokaryotes and eukaryotes, the main difference or setups of them is prokaryotes are typically haploid, usually having a singular circular chromosome found in the nucleoid. Eukaryotes are diploid. DNA is organized into multiple linear chromosomes found in the nucleus.